right, so we're going to take some time to talk about some non-Mendelian genetics now. So up until now, what has been discussed are Mendelian genetics. And what that is what we've really been talking about is called complete dominance. And this is called complete dominance because this is the situation where the heterozygote exhibits the trait associated with the dominant allele, but not that of the recessive allele. So we have the dominant allele, and then we have the recessive allele. And the reason this is complete dominance is because the dominant allele is completely dominant over the recessive allele. So what we would see in this case would be the big D, whatever this happens to represent at this time. So again, if we had the heterozygote, which is the big D, little d in this case, the big D would be the phenotype. Whatever big D stood for would be the phenotype. And then we wouldn't see any of whatever the little d coded for, whatever the physical characteristic would be of the big or of the little d. So this right here, complete dominance, would be Mendelian genetics. Because this is what Mendel was able to observe, because he wasn't looking at the actual DNA and wasn't able to get as many details. But what we know is that everything doesn't follow Mendelian genetics. So we're going to talk about some differences in Mendelian genetics. First, we're going to talk about codominance. So codominance is when the effects of both alleles are apparent. So the effects of both alleles are apparent to the fullest extent in the heterozygote. So the effects of both alleles are apparent to the fullest extent in the heterozygote. So meaning that one is not completely dominant over the other. So we see both characteristics, both alleles are present, and not in some sort of lesser way, but completely present. So one example of this would be blood types. So blood types. So in our blood types, what we have are basically our different blood types. We have the A and the B antigens that we see on our blood cells. So we have the protein products, our alleles will code for A or B proteins on the outside of our red blood cells. So what we see in the case of our blood type, we can actually have type A, and what this means is that we have a person that can be AA, and the way that blood type is written are these I's, and then the characteristic is up there. So it can be AA, or in this case, the way blood type is written, it can be A, and then this could be the heterozygote, which is written as a lowercase I in this case. So if we have type A, we could get the one allele from the mother, which would be coding for blood type A, and one allele from the father, which would be coding for blood type A. Or we could get one allele from the mother or the father, which codes for blood type A, and then we could get the other allele from either the mother or the father. And this little I, what this stands for is no proteins on the blood cell. So one parent would contribute a no proteins on the blood cell, and the other parent would be contributing A proteins on the blood cell. And so then in that case, what we would see would be A proteins on the blood cell. So that would be a type A, for example. Then we would have type B, and in type B we would see something similar. We could get B from the mother and B from the father, or we could get B from either the mother or the father, and nothing, or no proteins, the little I from the mother or the father. And then where we see codominance now is in type AB. So type AB then is where we get type A from either the mother or the father, and then type B from either the mother or the father. And so when we see type A, we have our red blood cell. And then when we have A, let's say we have little triangles. These are little A proteins on our A blood cell. So it looks something like that, let's say. If we have B, let's say that these are little kind of circles. So our B protein sticking off of our red blood cell looks something like that. So then blood type AB is going to have both the triangles and the circles, or the half circles here. And what that means, again, codominance means that in the heterozygote, so in one that has both, right, both the A and the B, we see both A and the B present. One is not lesser than the other. We see both. They are codominant at the same time. They share dominance at the same time. So that would be our AB. But then we also have type O. And type O then is actually just a little I, which then, of course, would be no proteins at all. So it would be a completely clear blood cell here. And then in that case, of course, we would have to get a little I from the mother and a little I from the father in order to have blood type O. So an individual that has one A and one B allele will then have our type AB, and this is where we see the codominance. So neither one is dominant. Instead, they show the full extent of their phenotype in that case. Another example that we see are in some flowers. So some flowers will have the white allele, so it will be a white flower, and then it can be crossed with a red flower. And then in that case, what we end up having are blotches of white and red. So this wouldn't be an example of it making pink, red and white making pink, and instead it would be blotches of white and blotches of red in the flower. We can also see this sometimes in cows. If you cross a white cow, one that's all white, with a brown cow, sometimes what you see are cows that have splotches, have different spots on them. And so in this case, the areas that are white on the cow is because they're showing 100% of the white color, and the areas that are brown on the cow are showing 100% of the brown color, and they are the heterozygote. They have the coloring or the allele for white, they have the coloring or the allele for brown, and so they're showing both. One is not dominant over the other. Both are seen. So this is codominance. The next type of non-Mendelian genetics that we're going to talk about is called incomplete dominance. So in incomplete dominance, we have an intermediate phenotype. So in the intermediate phenotype, as it implies, it is between the way the trait is expressed in the homozygous dominant or the homozygous recessive person or item. And so we see something that's between. So this would be an example of if we cross a red, I don't know what happened there, a red flower with a white flower. What do you think would be our intermediate phenotype? So in this case, we're not getting blotches of red and blotches of white, which is what we would see in codominance. Instead, what we're getting is some sort of intermediate, meaning an in-between phenotype. So in between red and white, mix those together, we get pink. Another example of this is sickle cell. What we see in sickle cell is the sickle cell allele shows incomplete dominance. So let's write that down here also. Sickle cell is an example of incomplete dominance. So the heterozygote actually has the sickle cell trait. This is actually shown as HB, and then they have the sickle cell trait, and then they have a regular blood cell. So the heterozygote has incomplete dominance, and so they have the sickle cell trait. So something that's in between a regular blood cell and a sickle cell blood cell. So if we did a Punnett square for something that's incomplete dominance, let's use our flower color. So let's say we use R, and R codes for red. And then we have white. So little r is going to code for white. And in this case, if we take our red and cross it with our white flower. A very simple Punnett square here. Then what we get is big R, little r, and in this case, because it shows incomplete dominance, this would be a pink flower. So in this case, if we are crossing the parent generation, the P1 generation is our homozygous for red and homozygous for white. So we cross the homozygous for red up here with the homozygous for white here, and what we get is 100% pink flower, or 100% heterozygote. Then, of course, if we cross that heterozygote with another heterozygote, so if we cross a pink flower with a pink flower, what we see in this case, if we do our simple Punnett square, is we see what we get out of the pink flower crossed with the pink flower is we get one red flower, we get two pink, and one white. So when we see incomplete dominance, what we end up with is if we cross our F1 generation, which is what we had here, when we cross the parent generation, we end up getting the F1 generation. The F1 generation is the pink flowers. If we cross our F1 generation, the pink flower with the pink flower, then what we get here is our F2 generation. Our F2 generation has a 1 to 2 to 1 ratio, and the 1 is our red flower, our 2 is the intermediate, and then the 1 is our white flower. All right, so now we're going to talk about the next non-Mendelian trait or characteristic. And the next one we are going to talk about is called pleiotropy. We'll just talk about this one briefly. What pleiotropy means is when we have one gene that has many effects. So in this case, what a good example for this one is the sickle cell example again. So the sickle hemoglobin is going to affect many areas of the body. So sickle cell hemoglobin. So the sickle cell is going to, of course, then make the blood cell shape like a sickle instead of a nice round kind of biconcave shape. And so when it is a sickle shape, then it's going to affect various areas of the body and actually causes some serious issues. So it can cause things like kidney damage, it can cause lung damage, it can cause brain damage, causes pain, can cause anemia, difficulty breathing, all sorts of different characteristics. So in this case, we have one single gene, and this one single gene is going to have many effects throughout the body. And that's called pleiotropy, and that's, again, something that's non-Mendelian. So Mendel didn't see that he could look at one single characteristic or one single allele or one single gene, and then that would have effects over the entire organism. So the next principle that we're going to talk about is called multiple alleles. So in this case, this is when, as its name implies, when we have 3 or more forms of a gene, and they exist across many people in the population. So 3 or more forms of a gene. So the example that we used earlier, our blood type, is another example, or is a good example of multiple alleles. So in our blood type, remember, we had A, B, and then we had O, or the little I. So this is a good example of multiple alleles. In this case, we have an allele that codes for A, we have an allele that codes for B, and we have an allele that codes for lowercase I, or O in this case. And so one person can only have 2 of these alleles, but there are 3 available. So we see differences throughout the population. So again, what we saw using those is instead of seeing just A, or just B, which would be 2 alleles, or A, B, which gives us kind of our three options like we've seen previously when we have, the uppercase letter and the lowercase letter. We end up having three different options, basically. In this case, then, we have a fourth option, which includes the O, or the third allele in this case. And we see this very, very clearly in things like flowers. In flowers, we can have a rose, for example, and a rose can be red, it can be pink, it can be coral-colored or yellow or white. So there are many, many different alleles that code for flower color, for example, or rose color, for example. We see the same thing when we see eyes in the Drosophila fly, for example. So they have different color eyes. It's one gene, not for humans, but one gene for the Drosophila fly, which is a fly that's used in experiments often. And so there are many, many different forms of that gene, multiple alleles that can code for that. Now, talking about eye color, we can talk about eye color in humans, which is not a case of multiple alleles, but in this case what we're talking about is polygenic inheritance. So polygenic inheritance is when we have variation in a trait, so something like eye color or height, and it's independent of environmental influences. So first, variation in a trait, not due to environment, and then it involves two or more genes to determine the trait. So two or more genes to determine the trait. So what we've been talking about up until now is we're talking about one gene, and you can have different forms of the gene, which are called alleles. So let's say, for example, we're talking about our multiple alleles, and we're talking about blood type. We're talking about the blood type gene, and in the blood type gene, you can get either A or B or O from your mother and from your father. But we're still talking about the blood type gene, and that codes for the blood type, which codes for the proteins on the red blood cells. In polygenic inheritance, what we're talking about are two or more genes that are going to determine the trait. So, for example, eye color in humans. Eye color in humans is not just so simple as having an eye color gene, and you get one eye color gene from your mother, one eye color gene from your father. In this case, it incorporates several genes. So you'll have several different alleles that you'll get for eye color from your mother, several different alleles for different genes that you'll get from your father, and then that will contribute to eye color. Another example is skin color. So there are many, many different variations for skin color in humans. And so there are multiple genes that are going to determine the trait of skin color. So you'll get multiple different genes or alleles from your mother, multiple different alleles from your father, and the combination of all of these different alleles on these different genes are going to contribute to skin color. Another example is height. So there's lots of variation in height. And so in height, we have multiple alleles from mom, multiple alleles from dad that are going to contribute to the height of the organism. So a lot of times the trait that we see in polygenic inheritance, so those that are produced by polygenic inheritance, tend to be in bell curve distribution. So if you think back to some math here, we can say these are the number of people that have something. And then this would be the characteristic. So we could say height, for example. The characteristic of height. And so a bell curve looks something like this. And so we see people that are down here, which would be very, very short people. Over here would be very, very tall people. But most people lie somewhere within here. And pretty much in the middle there, an average height is what we see for most people. And this is what we would see with polygenic inheritance. So most individuals display near an average trait value, kind of toward the middle, rather than the extreme, so on the short end or the tall end, for example. So that's going to be our polygenic inheritance. The next characteristic that we're going to talk about is called linkage. So in linkage, this is when genes on different chromosomes, if you think about genes on different chromosomes, they're sorted independently. Right, so this is Mendelian genetics. Talking about Mendelian genetics, what we have already learned is that when we have genes on different chromosomes, they sort independently. Right, so meaning that one gene on chromosome number 2 isn't going to always go with the gene on chromosome number 4, for example, and you'll always get 2 and 4 from the mother, and they're the same. That's not how that happens. So they're sorted independently. Sometimes you get a particular allele or gene from chromosome 2, but you'll get a different one on 4. Chromosome 4, you'll get a different one on 2. So they line up independently on that metaphase plate. But genes that are close together on the same chromosome are linked. So the traits tend to show up together. So when we're talking about linkage, we're talking about genes that are close to each other on a chromosome. And to show up together. So, for example, the genes that control skin color. So, for example, the genes that control skin color tend to be close to each other, and so some those genes tend to show up together. Also, things like red-haired people that have red hair, tend to have freckles. And this is because those genes are very close to each other on the chromosome, and so they tend to show up together. So if we look at our chromosome for just a moment, we can kind of take a look at what that means. So what that means is, let's say this right here is the gene for red hair, and let's say this right here is the gene for freckles. Even if we are going through meiosis and we do some crossing over, the chances of the crossing over happening between those two genes are very, very slim. It, of course, does happen, but oftentimes they will cross over either above the two or below the two, just because they're so close together. Now, if they were further apart, then we might see those separate more often, but since they're very, very close together, oftentimes the two genes will go together, and so we see those, and that's called linkage. They're linked. So red hair is often seen in people, or freckles are often seen with people that have red hair, or the genes that control skin color and freckles in this case are very, very close together. And so that would be called linkage. So again, we have different chromosomes are going to independently assort, but when they're close to each other, even when we have that crossing over, a lot of times we see the same genes kind of go over together. The next principle we'll talk about is environmental effects. And so in this case, as the name implies, we're talking about the environment and the effects the environment can have. So the environment can cause different phenotypes from the same genotype. So the environment causes different phenotypes from the same genotype. So some of the environmental effects when we talk about, maybe not necessarily humans all the time, but we can talk about plants. If you put different plants in different temperatures or different organisms, there's bacteria, Serratia marquescens. If you grow it in a certain temperature, when it grows, it's a big colony that looks very, very red. If you grow it in a different temperature, it's a big colony that looks cream color. And that's simply due to the temperature that it's grown in. Other things like pH can affect things. If you have a different pH in different flowers, they may have different colors. Different amounts of water given to the plants or different nutrients given to the plants or animals. Different types of sun exposure. So different tan. So you could have twins, for example, that have the same genotype. They have the same DNA, identical twins that have the same DNA, but if one tends to spend more time out in the sun than the other, then their phenotype, their skin color, looks different than the other one. That's just due to sun exposure, the environment. So that would be changing the phenotype. The physical characteristic of their skin looks different than their identical twin because the other identical twin, for example, stays inside more often. Another example is the Arctic fox. The Arctic fox has a gene that produces the pigment in its fur, and when it's in a warm climate, then it's going to be a red-brown color. But when it's in a cold climate, then it actually turns the color of the fur white. So this is great for the Arctic fox, so that when it's warm, and we have all of these different colors of plants around, then they can hide in the plants because they're this reddish-brown color. They can hide in shadows and such. But in the winter, when there's lots of snow around, then its fur changes to a white color, and then it can kind of blend in the white color. Another example are different flowers, like I mentioned. If the soil is acidic, so hydrangea flowers, for example. Hydrangea flowers. If they are in soil that is acidic, then they end up being blue. But if they're in soil that's basic, then they end up being pink, for example. And then I already mentioned the identical twins. For example, differences in nutrition could make one shorter than the other. If one gets a lot less of the nutrients, they may be a lot less healthy. Or tanning, being in the sun exposure, might change the color of the skin, so that would be different phenotypes rather than a genetic difference. All right, so now we're going to talk about something called sex-linked or X-linked inheritance. So X-linked inheritance. So really quickly, our sex determination, some background, is when we have an XX, our last set of chromosomes, our 23rd pair, if they're an X and an X, then we have a person that is female. And if they are XY, then we have a person that is male. And the reason for this is because the female actually inherits an X chromosome from the mother and inherits an X chromosome from the father. And an X chromosome is a full-length chromosome. In the male, the person inherits an X chromosome from the mother, but then they inherit a Y chromosome from the father. And the Y chromosome is a shorter chromosome. It holds less alleles, less genes than the X chromosome. So the Y chromosome right here is much, much smaller. So obviously it can't hold as many genes. So it carries fewer genes. And most of the genes on the X chromosome have no corresponding alleles with those on the Y chromosome. So let me write that down. So most genes on the X chromosome have no corresponding alleles on the Y chromosome. So in this case, these are known as X-linked genes because we have them on the X chromosome but not on the Y chromosome. And so then we have different characteristics that can be carried from generation to generation on the Y chromosome, but then we're not getting something from the X chromosome. So in this case, we have some genetic disorders that are X-linked, and these are often caused by recessive alleles. Because if someone displays the dominant allele, so if you display the dominant allele, or if you have the dominant allele and you, let's say, go back on that, if we display the dominant phenotype. So someone who displays the dominant phenotype but is heterozygous for the trait is going to be a carrier. So if we think about the X-linked in this case, if we talk about genetic disorders that are caused by recessive alleles, that's because if we have an X and an X, right? If this first X is somehow has a disorder on it, right? Let's say it has some sort of genetic disorder, the person will still have the other X that may be able to make that particular protein. Let's say the disorder is on this X, that gene had a mutation in it, so it's not making the correct protein. But the person has another X, and that X still codes for that same protein because it has the same genes on it. So they can rely on that second X in this case in order to make that particular protein. Now, if a person is XY and this X has that mutation, then on the Y chromosome, they don't have that same gene. And so in that case, they will end up having the disease. And that's why we see this genetic order is caused by a recessive allele. So if the person, or in this case, if it's XY and XX, so in this case, let's say the person has the dominant phenotype, which is the appropriate code for that protein, and then this one here has the inappropriate code, then it's dominant, right? So this X is dominant, which is basically what we set up here. I just didn't use dominant and recessive. So we have the dominant or the non-mutated version of it. But then over here, if the person on their X has the dominant, then they're fine. But if the person over here doesn't have the dominant, they have the recessive, then they're only making the recessive, so only making the mutated protein, and then they have the disease. Now, in this case, the example that I just showed here, this person is actually what we would call a carrier. So in this person, what I was writing here, this person displays the dominant phenotype, right? So they have the little d here, which we're just saying is the mutated gene, the not good one. But they have the dominant one here, the big D, which is going to be coding for that particular protein. So they're going to have that protein. So this person, you might not be able to even know that they have one allele that is mutated because they're relying on their dominant gene to make whatever protein it is. And then the person is living a normal life, not knowing that they have this genetic disorder. But this person is called a carrier. And this is because they have the recessive allele, and then they're carrying that. And then that can be passed down to the next generation. So let's put some actual information to this dominant and recessive. So one of the common disorders that is caused by an X-linked recessive allele is red-green color blindness. So with red-green color blindness, so in this case, if you have XX, right, and again, if you have one that codes for color blindness, in this case, let's say this one codes for color blindness, then the female has this one to rely on, and then they can still see red-green. In a person that has red-green color blindness, it's difficult to tell the difference between red and green. There are different versions of this, different variations, but a person will not see red or green. Sometimes they can see different variations of gray, so they know which one's red and which one's green, but they don't actually see the colors red and green. And so in this case, what we're talking about, we can say that in red-green color blindness, the recessive allele is going to be on the X chromosome. So our alleles, let's say, instead of using these X's, let me get rid of this here. Instead, let's just use some R's. So we'll just say R is normal vision, the big R, normal vision. We'll say the little r is colorblind allele. And then on the Y, we have no allele, because this is X-linked again. Put that up here so we remember. So this is X-linked. So we have no allele for red-green color blindness on the Y chromosome. So the different genotypes that we could have then are different genotypes. We could have big R, big R. We could have big R, little r. We could have little r, little r. We could have big R, Y, little r, Y, and then we could have Y, Y. In this case, not really, because this is lethal. And so a person isn't actually born with two Ys, so that ends up not being an option. So then we end up with our phenotypes, and then our phenotypes will be a normal female, so normal vision female, and that would be big R, and big R, little r, because even though we have the little r, the big R is going to take over and allow for normal vision. We could have a colorblind female, red-green colorblind, of course, and that would just be little r, little r. We could have a normal vision male, which would be the big R, Y, and then the colorblind male, which would be little r, y. So you can maybe see here that there are two options for normal vision, normal color vision in females, and only one, which is the recessive allele for colorblind female. Versus in a male, we kind of have a 50-50 shot here. If we have a parent that has an X chromosome, if the mother has a recessive allele here. So we actually do see red-green color blindness more in males than in females because of this. So if we did a cross really quick, if we took a colorblind female, so a colorblind female, cross that with a normal male, normal vision male, what would our letters be here? Right, little r, crossed with big R, Y. So our cross then, if we did our Punnett square, let's say we have little r, big R, Y. So if we did our cross, then let's see what we end up with. We end up with our genotypes. Our genotypes are big R, little r, 2, and then little r, y, 2 of those. So it's a 50-50 shot here. If we have a colorblind female crossed with a normal male, we get 50-50. 50% that we're going to have a big R, little r, which, what does that mean? So the phenotype, this would be 2 normal female, right, because we have the X and X, and 2 colorblind male. So in this case, if we have a colorblind female, if there's a female that's colorblind, then if they have a male child, then all male children will be colorblind. If they have a female child, then all female will not be colorblind if they're crossed with a normal male. Now, if we had a normal or a, let's say, hybrid female, so by hybrid, what I mean is that they are a carrier. So let's say the female is a carrier for color blindness, and we cross that with, let's say, a normal male. So in this case, what we're saying is that we have a female, let's get rid of the word hybrid here. We have a female that's a carrier, so she may have no idea that she's been carrying red-green color blindness. So in this case, the female carrier would be, she would have one normal X and one diseased X or colorblind X. And then we're crossing that with a normal male. So the normal male would be a big R and a Y. So we have two parents, neither of them are colorblind, and perhaps the female doesn't even know that she's carrying colorblindness. So if we do the cross between that, we see are very different odds here. So up here, we get a 25% big R. We get 25% 1 big R, little r, 25% big R, Y, and then 25% little r, Y. So then what this means is that in this case, big R, little r, we have a normal vision female. So all females born to a carrier female and a normal male will have normal vision. But in this case, we have a 50-50 chance here. We have a normal vision male, and we have a colorblind male. So this is how sometimes females will know that they are carriers, is because if they have a son that is colorblind, they may have had no idea that they were even carrying this trait, but if they have a son that is colorblind, then that means that they themselves have passed on that colorblind allele. And then they know that they have one functioning allele because they don't have color blindness, and then they have one allele that is coding for color blindness. And so this would be an example of our X-linked inheritance. So it's on the X chromosome. The Y chromosome doesn't have anything that can compete with it or cover it over or dominate over it, and so then that's going to be passed on. Whichever X goes to that person, they will show that. So it ends up being a recessive trait that gives that genetic inheritance. So this kind of summarizes all of our non-Mendelian inheritance. So we just talked about our X-linked inheritance, our different environmental effects. So the same genotype can cause different phenotypes. We talked about linkage, where our genes, if they're very, very close to each other, tend to show up together, like red hair and freckles, for example. We talked about polygenic inheritance. So this is where we have variations in a trait. So that involves 2 or more genes that determine the trait. So, for example, height or skin color or eye color in humans. Multiple alleles, a good example of multiple alleles are the blood type alleles. You should be able to do a Punnett square with our blood types. So determining a blood type, if we have a person that is a blood type A, you have to take into consideration that they could be AA or they could be AI, for example, and then doing crosses based on that information and determining the outcomes for that. Then we talked about pleiotropy, where it's one gene that has many effects. The example we used there was the sickle cell gene, affects a lot of other things in the body. We talked about incomplete dominance. This is where it's incomplete and has an intermediate phenotype. You can remember the incomplete is intermediate phenotype, so something in between, so red and white is pink. We talked about codominance is the first non-Mendelian trait. So codominance meaning both are dominant. So co meaning sharing, and then dominance. They share dominance. We see all of the protein A and all of the protein B in blood type AB, for example. And then the first thing that we talked about was called complete dominance. And complete dominance is just what we've been talking about previously, or what you've learned previously about non-Mendelian genetics, or about Mendelian genetics, rather. So Mendelian genetics is based on dominance. There's a dominant allele and a recessive allele. And then in our heterozygote, the dominant allele is going to be the phenotype compared to the recessive allele. So that's what we had talked about previously. And now we just went over all of these different characteristics of non-Mendelian genetics that you should be familiar with.